So that'll kind of be where we're going to start, where we picked off. We were talking saprotrophs last time, I think, right? Organisms that basically live on dead and decaying matter. And I think we had a quick chat about Chernobyl, which I tell you, I would go there. It fascinates me if I was allowed to travel. Monday marks 21 days since my first COVID shot. So I will be back in the city on Monday getting number 2. I'll definitely report back then. But I'm hoping that that will open up travel for me, for us, or at least make it a little less risky to do that. I don't know if folks have been paying attention. Out in Hawaii, they're doing what's called a digital vaccine passport. And I'm sure folks haven't seen that because Hawaii is probably the furthest thing from all of our minds. But in order to fly between islands, you need to be vaccinated and you have to be able to prove it. And that will circumvent the PCR reactions that are needed to not quarantine. And so obviously, if it works for the Hawaiians, I would fully expect the rest of America to go that route, notwithstanding legal challenges, of course, to return to some semblance of a new normal. And that's promising, right? First shot, we're seeing reductions in transmission, which is good. People weren't sure about it. And we're seeing now the guidelines haven't been increased, but we're seeing protection now in month 4 for people who are vaccinated early and who are in trials. So while they're saying only 3 months of not having to be tested, we're already getting data for month 4 and month 5. So watch those numbers go up. Let's see, news-wise. Obviously, COVID's all over the news, but there's a little sneaky that I want to put in the chat that's related to influenza. And that is, there was just a new strain found, which starts with H5. And the crazy thing about H5 is the last time we saw an H5N1 strain was an avian flu that had a 60% mortality rate. 60% mortality rate. And when I was just starting at the college in 2006, we had thought that that could have been the next great pandemic. And we were told that public safety had body bags for all of us as faculty and staff because they were expecting it to be that bad. And of course, it came and it went and nothing really happened, right? And then we had the H1N1 in 2009, came and went. I think 150 people passed. We moved on. And it wasn't until Bill Gates in his TED Talk, until what, that was 2015, 2016, did the idea of, hey, there's a pandemic brewing come back out there. H5N8, I've never seen. I don't know that anybody's ever seen it. And the people that it was reported in were asymptomatic workers on a farm that were handling birds. So not to take things at face value and run with it, I want to know why they were checking these people and how this variant was discovered, right? Because if you're healthy and feeling good, you don't test somebody for something that doesn't exist. So obviously that's of concern. And H5N8, I've never even seen the N spike in 8 form. Now, H and N go from 1 through 15. So there's quite a few combinations you could have at each spot. H3N2, H1N1, H2N1 you see, H12N7, H12N9. You see a lot of these. H5N8 is brand new. So that was a little gem hiding on the New York Times site. For what it's worth, I'm just mentioning it. I don't know what it's going to do, if it's going to do anything, but we're so focused on one. Let's not forget that there's the seasonal stuff going on that we should still pay attention to. Sure, no one else was probably looking at anything other than COVID, and I get it, right? That's dominating the news. There is a New York strain that's out there. I don't know if anybody caught that. They found it in basically all of the boroughs in the city, which means it's out there. And so that one apparently looks different than even the LA or the England strand. And so that bears watching as well. So there's a lot of health stuff going on out there. And I'll tell you, we're literally living through history. And so as my grandmother used to tell me about polio, and I'll clean this up because the language was not appropriate, and the things that went on in New York City in the 30s, 40s, and 50s, honestly, we're learning about COVID and the things that are happening in America in the 20s, right? And it's ironic that it's 100 years apart. And it's ironic that there's going to be a sense of nationalism after a shattered economy. So, you know, the old saying, history repeats itself. It's time to understand it. And I think scientifically, we're doing a good job. It's going to be our job as scientists, healthcare professionals, people in the know to get the information out there. You're not making choices for folks, but we want to educate, right? And that's the business that all of us at some point, whether it's me as an instructor or you guys as healthcare folks, right? We're all in the ignorance business, trying to get our clients and our patients to understand what's going on and how to live better. And so I think that's a fair place to kind of start the evening, right? We're all in the ignorance business, and we're just trying to bring folks up to speed. So let's see. Some of this is going to draw on A&P, and I'm assuming that some of this tonight will be kind of easy to think about. Some of this may be a review. I may ask folks to kind of help out and chime in, especially if this is stuff that you remember. And obviously, we've talked about this. We've talked a lot about this. And so I just want to have this up on the screen to make sure that we remember osmosis and diffusion, right? So these microbes don't have hearts generally. I should say generally. When we get to the helminths, there are pumps in these worms. So we can call them a heart, sort of. But the idea that most of these organisms in the media that you provide, or in the patient's blood, or in a sputum sample, is relying on diffusion and osmosis to get what they need. And so if they're an aerobe, to get oxygen. If they're an anaerobe, to get their carbon dioxide and whatever else they're fermenting. We can do this through diffusion, which is passive, not requiring ATP, but it does use energy. And I think we might have touched on that last time. If this was a chemistry class, we'd call this Brownian motion. And so Brownian motion is really at the speed limit of physics in the given area. So the speed limit is slower in a solid than it is in a liquid, than it is in a gas. And so the higher the temperature, generally the greater the transport. The higher the temperature within its range, and we'll talk about those ranges, generally the faster the metabolism is. So there are correlations here. If we are speaking chemistry, diffusion is the transport of solute from a high to a low concentration across or along a gradient. For us, obviously, we're talking about food. We're talking about chemicals. We're talking about particulates, not necessarily solute per se, but that is the right definition. And osmosis is the movement of solvent from a high concentration to a low concentration along a gradient, or really across a biological membrane if we want to be more specific. And that membrane, as we remember, is semi-permeable. So there's a couple things that have to hold true for this stuff to move. And for our cases, are we really going to talk about solvent? Probably not, right? We're more interested in water. And so both of these processes do not use ATP. They occur as long as there's a gradient and as long as the molecules meet the polarity requirements, the size requirements, the receptor requirements. So as long as the size is right and the polarity is right and the channel is open, the likelihood of movement is pretty good. That last one, and since I mentioned channel, it's a good lead-in. Facilitated diffusion or facultative diffusion requires a protein, right? It requires an extra little something to get it into the cell. So things that are too big or things that are charged inappropriately, things that are polar, proteins, small peptides would need a transporter in order to get in. You would see the gradient, but you would say, wait a minute, this stuff is too big. Wait a minute, this stuff is polar, but wait, it's getting in. Kind of as your clue. The active transport stuff generally requires ATP. That's the active part of it. Largely, there's carrier proteins involved, especially when you're pushing against a gradient. So we're going from low to high, or the equivalent of pushing a ball up a hill. One of the good examples here, probably from A&P, would be something to the effect of your sodium-potassium pump, right, where potassium is pumped actively against a gradient. Changes in the plasma membrane, in particular with exocytosis, endocytosis, phagocytosis even, would probably require amounts of ATP that would be able to be measured. And we throw those under bulk transport because it's not just a molecule, it's bunches, groups of molecules, maybe even an organism, depending upon what we're looking at. So most of that should be review. If there's concerns or anything about it, just toss that in the chat. But this is ever so important for microbes because they are one cell thick. They do not have a pump. They are pretty much in constant contact with the environment. So even the diffusion rate in a liquid, which is around 1 millimeter an hour, is more than sufficient for these microbes. Now, when you think about exoenzymes and the saprotrophs that we left off with last time, that means they have to diffuse their enzyme out of the cell. It's going to move on a millimeter an hour, give or take. It has to work on the substrate, right? So the food it tries to digest, and then it has to diffuse back. So the process isn't a very quick one, but for things that are too large to bring into the cells, it's the one that they have. And let's see, so a couple of pictures just so we can see, right? You drop a sugar cube into your coffee or tea. I don't know if you guys use sugar cubes. It's weird, right? In America, you don't see it. I was in Ireland, that's all they had was cubes. And the important thing here is when you look at what's happening, that's the sugar cube in the bottom right corner. These are the molecules that are coming off, right? And without artificially increasing the diffusion gradient, it's a long and slow process. So when I say artificially increasing, I mean the tea is hot, so the molecules want to move faster. You're probably going to be served a spoon with this. So you're going to stir, and that's going to help add kinetic energy to the mix and move things around more quickly, right? So there's ways we can speed this up, but if you leave it alone, you're probably going to see that cube in there for several hours while it dissolves and while the molecules start to move around. And obviously, supersaturated, undersaturated, saturated are things that should be pretty good in terms of the carrying capacity of the solvent related to the solute. In the body, this is important. I'm sure in A&P you learned about gout and you learned about kidney stones and those types of things, supersaturations of chemicals. So the stuff that you've learned there certainly works here in the solutions or in your nutrient media, if you prefer to go that way. I don't know if we're in the more is better mentality, but that's certainly not true. And I can give you examples of things like ammonium toxicity, right? Nitrogen is good, too much nitrogen is bad. And so we still want to make the medium appropriately with the right concentrations of things. Osmosis here is another one, and we're seeing something interesting here. And this is one I might ask for a little help on, because if you notice, we're talking about osmosis. We have a gradient, and we should see an equal and opposite reaction in order to reach an equilibrium. What we're instead seeing is that water moves one direction, but the solvent doesn't move the other direction. But the solute, excuse me, the little orange dots don't move in the other direction. And so maybe we could work up a hypothesis as to what's happening and why.
The water moves to the side that has more of the solvent in it.
All right, so let's break that down. The water moves to the side that has more solvent in it. Is that what you wanted to say? So osmosis is the movement of a solvent along a gradient from high to low. So if I give you that definition, does that make sense?
The solutes can't diffuse through the membrane.
Right, so the solutes cannot. They want to, but they're unable to because, I don't know, they're probably too big, right? So the solute wants to move to the left, which means the water wants to move to the right, or in this case, right, the solvent. So there is a gradient when it comes to solvent concentration. Now, with relation to solution X and Y, maybe it could be explained in terms of, and I know you're going to cringe when I say this, hypertonic and hypotonic. Oh, those are good words, aren't they? So the solution on the right is hypertonic to the solution on the left. Are we comfortable with that statement? Right, the solution on the right has more solute than the concentration of the solution on the left. So if it has more solute, then it has less solvent, right? And we're doing this out of 100%. The solution on the left is hypotonic to the solution on the right. The solution on the left has less solute in it than the one on the right, which means it has more solvent. Right? I know these are dirty words, and so what that means is the water should osmose from left to right because there is a higher concentration of water in the solution on the left than there is on the right. And so what you're seeing in the bottom pane is the displacement of water changing because the water moved across the membrane even though the solute could not. And the solute's probably too big, like sucrose or something. right? And obviously there's osmotic pressures and there's things of those nature that are at force here. But just in terms of osmosis, this is the demonstration of the movement of solvent, of water, from high to low, reinforcing those things like hyper and hypotonic that you've seen in nursing and calculations and drug administration in Chem 105, in A&P. The idea here that obviously if you are caring for a patient, you want to give them something that's isotonic where at all possible, so that you add volume, but you don't change concentrations. When you put red blood cells into hypotonic solutions, they explode. When you put red blood cells into hypertonic solutions, right, they shrivel or they crenate. All right, so good review nonetheless, but what works for us works for the microbes with one caveat, right? They have a cell wall. So they don't necessarily explode, but they certainly have water moving, and they certainly could become very, I don't want to call it turgid, that's a plant word, but pressurized. There could be a high amount of water relative to solute in a bacterial cell without it exploding. So in our bodies, if we think about it, that cell wall keeps them from exploding. But in the case of you give an antibiotic to a patient, like a beta-lactam or a cephalosporin, you undermine the integrity of the wall, and osmosis does the rest of the job for you. The water rushes in because the cell wall is made poorly, and the cells lyse. Right, so there's good background here. We just have to kind of remember what happens to red blood cells and then kind of apply it to bacterium and then throw in that antibiotic, which is kind of a cool thing when we think about it. So should we ask questions on this, or is this like enough of a flashback from A&P that we don't want to touch it? That's always a loaded question, I assume, but I'll ask anyway. And I'll keep my eye on the chat just to see. All right. So there's a bunch of other stuff in here, right? So the idea of gated channels probably is okay with folks. So some channels are open, some transporters are open, some of them are gated. And so the ones that are open just allow osmosis and diffusion to happen all the time. The ones that are gated usually open and close in response to a stimulus. And more times than not, those are probably more active processes, but it really depends on the situation we mean. So in this particular case, you have a protein molecule that's allowing whatever the green solute is to go from one side of the plasma membrane to the other along a gradient. And you'll notice that once there's some sort of equilibrium, that channel closes, which maybe isn't the way that we should be explaining it, but at least it's what this is showing on our graph. There's a bunch of other versions. Membranes have proteins. You're seeing kind of a receptor-mediated one in the top. You're seeing proteins help move things through on the bottom. So there's a lot of perturbations of this. The receptor one we're going to come back to heavily when we look at viruses. And obviously receptors need to match when a virus and a eukaryotic or prokaryotic cell come into contact. Here, whatever that molecule is, has to activate the receptor. And so that thumbtack looking molecule happens to fit into the recognition site. And you can think of it as a lock and key. That's totally fine. And once it fits, it actually moves through. The other version of this, and I guess I don't have a picture of it, would be instead of the receptor actually being a part of the gate, the receptor would be on one side or the other of the protein. Think of it like a neurotransmitter, the way a neurotransmitter would work. So if acetylcholine is released by the synaptic terminal and it hits the receptor on the postsynaptic neuron, it lands typically on either side, opens up the proteins, and then your ions move in. So there are a variety of ways it works, but those are just 2 or 3 of them. And you know, the lock and key method pretty much explains most of it. And then obviously phagocytosis is about organisms or particles. Pinocytosis is about liquids or dissolved items, solutes or ions, proteins, what have you. And we're bringing them into the cell. And you'll notice here that they're coming in as vesicles. Not always the case, but certainly in our protistas and our fungi, this would be applicable. And then the reverse of this is exocytosis. So a vesicle would merge with the membrane and kick it out. So you're seeing pseudopods or false feet you're probably thinking like amoeba or paramecia or euglena or things on that order. Interestingly enough, you know, phagocytosis is one of those things that our immune cells use. And it's interesting to watch macrophage phagocytize tuberculosis, M. tuberculosis. And it's funny because the macrophage do a really good job at engulfing it and bringing it into the macrophage. But because of the mycolic acids, they're not able to hydrolyze it. And so that mycobacterium actually lives inside the macrophage, dividing ever so slowly. And what you hope the body does is wall it off or form a tubercle. And so while it's good that it's walled off and it's good that it can't escape, how many tubercles are you able to have in the lung or in the body before the tissues that they reside in fail? The other strange thing about the tubercles is they're walled off, they're kept at bay, but with tuberculosis, we see reactivation. So years down the road of an active case, Eleanor Roosevelt's the one I really like to use a lot. So Eleanor Roosevelt, wife of FDR, lived, born, raised, died in Poughkeepsie. And the cause of death is actually listed as tuberculosis, even though she was completely fine until a car accident reactivated the TB. So she was in a minor fender bender in Poughkeepsie. Maybe she struck the steering wheel. Maybe she didn't have her seatbelt on. It's really hard to say. But she, I guess, inadvertently opened or burst or popped one of those tubercles, allowed some of the mycobacterium to escape. And at her age, I want to say 67 or 70, maybe even more at the time, her body wasn't able to wall it off the way it originally did. So when we look at transport, the movement of these particles is really cool at the organism level, at the ecosystem level, because I guess humans can be an ecosystem. And at the immune system level, it's fascinating how the process is the same, but there are so many different outcomes that you can look at with one single process. Some of that is actually phenomenal. So I'll give that a pause. We will pick up with this, but I'll give that a pause and just see how we feel about transport, how we feel about passive versus active. I don't know that I want to ask the dirty question, but I will anyway. Hypertonic and hypotonic, right? Obviously refer to the amount of solute in a solution, and it's always the amount of solute in a solution relative to another, right? I think that's the important thing. I can tell you my Gatorade, I don't know if you can, oh, there we go. Gatorade is hypertonic, right? So the first thing you should say is to what, right? To my tap water. Okay, Gatorade is hypertonic to my tap water. Well, that makes a lot of sense, right? Reading the label here, it says, God, I wish I could read this. Sugar, dextrose, wish I didn't read this, citric acid, salt, sodium citrate, blah, 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 right? So there's lots of solutes. If I grab my seltzer, right, which I've got plenty of beverages, and trust me, they're all non-alcoholic, right, vintage seltzer. This one just says 0 for everything. There's carbonated water and there's natural flavor, even though, and I wonder if you can see that. Nope, I'm gonna kill the background, so don't laugh at my mess. Right, even though, nope, video, background, sorry. Even though it says on that top there, 0% juice. I don't know if it's gonna resolve, but that actually says contains 0% juice at the very top, along with every 0 on Earth here. So I guess I'll ask 2 questions here. My Gatorade is definitely hypertonic to my seltzer, right? It has more solute in it than my seltzer. Obviously, the seltzer is a bunch of zeros. It also means my seltzer is hypotonic to the Gatorade. We're okay with that? The seltzer has less solute in it than my Gatorade, right? And at all the zeros, that's pretty clear. So why did my seltzer company bother to put 0% juice on it? Does that mean there's no juice in there? Just seltzer?
Sugar, maybe?
No, there's no sugar in this. It's literally 0 fat, 0 sodium, 0 carb, 0 protein.
Because the picture on the outside. Just to cover themselves.
So So, you know, what if there, and this bottle, by the way, is 1 liter. All right, so 1,000 milliliters. How much juice could I actually put in there and still legally say 0%? 1 mil, 2 mils, 3 mils, 5 mils, right? Because 5 mils in 1,000 is 0.5%. Right? So they're not lying. They're not really being dishonest. There really is 0% juice in there. Lemon lime, I think, is what's in there. But there's only 4 mils or 3 mils per 1,000. So they get away with saying 0%, which doesn't mean none, right? That's the point I'm trying to make. It doesn't mean none. It just means there's very little in there. So for what it's worth, people always shudder and they say, well, I don't understand. Why did they just say juice free? Because it's not. And I've been the victim of this, so don't feel bad. I was in the Bahamas. I have to watch my salt. I bought a bottle of water that said sodium free, which was a misnomer because it had 0% sodium on it, but it also said 5 milligrams on the bottle. So there was a whole hot mess of advertising, which the only thing they shouldn't have said was sodium free, even though the 0% and the 5 milligrams were on there. Zeros are good. Yeah, I don't disagree. Zeros are pretty good. I love the stuff. And even if there wasn't any juice in here, it was just a regular seltzer, it's still better than a lot of things. And I don't worry about the CO2 because, as you're also aware, the carbonic acid in there is a buffer. So as you guys consume seltzer, it's mildly alkalizing, and it actually helps add bicarbonate to the blood. All right, so again, for what it's worth, just useless, not useless, random useless chemistry information. All right, so the stuff here that would be of interest to folks, especially if you guys felt the lab was a little wacky. Temperature requirements, oxygen requirements were the two big things. The nutrient requirements, there's a spectrophotometer in here, so I want to talk about those when we get there. Radiation's an interesting one, but let's face it, there are some organisms that love radiation. They would probably be extremophiles. You would probably call them an archaebacteria, like Adeinococcus radiodurans. And then there are organisms that love barometric pressure. We call them barophiles. And those would be things that you would find deeper in the earth. So I think the Soviets, and then continued by the Russians, dug the deepest hole ever into Earth. I want to say it was 37 kilometers or something like that. And so obviously samples that come up, if you actually want to study the microorganism, have to recreate not only the temperature and the oxygen requirements, but the pressure requirements. Otherwise, you could kill the organism. People who do ice coring, barometric pressure is a big deal. And unfortunately, when we do this, we're not necessarily trying to culture. We are more apt trying to figure out the chemical signatures to describe life on Earth at a time and space, rather than try to culture the stuff. I would be leery about culturing the stuff anyway. It probably hasn't seen the surface or the light of day in many, many, many years. And I hate to be the guy that unleashes a plague on humanity. But anyway, these are the things that we think about. I'm also going to throw gas in there. Obviously, oxygen is one thing, but there are things that love carbon dioxide. They are called canophiles. And so if you have a canophile, the amount of CO2 in the air is not enough. And so at times you have to supplement. If you grab something from a hydrogen sulfide field, you probably have to feed it hydrogen sulfide as a piece of its nutrient requirements. And the last weird thing I'll mention, and I really didn't include it in here because folks would think I'm crazy, is light. If you're something that's photosynthetic, if you're an organism that relies on feeding itself and it doesn't have light, then it doesn't grow. And so for mathematical models, it shouldn't surprise us, but it's weird for us to think of it this way, as light as a nutrient. So when we look, well, we're not going to do it, but if you look at Monod kinetics, or you look at growth curves and the formulas behind them, they'll treat light as a nutrient the way we treat nitrogen and carbon, especially if it's a photoautotroph. And so sometimes we probably need to consider light requirements here because our protistas may be photosynthetic, certain bacterium certainly are. And so I'm just mentioning it as an aside. It's not the main point of what we're looking at, but it does exist. So I figure I'll mention it. So we might as well start with temperatures. And I know, yep, cyanobacteria being one of them, right? The blue-greens have phycoerythrin, they have phycocyanin, and so some of them have chloroplasts, some of them have phycobilisomes. Same basic idea, pigments that use light energy to make chemical energy. So in cyanobacteria, which is a great example, especially if you look at the Hawaiian bioreactors, so they grow in these clear, fluorescent, light-looking tubes with bubbles. Obviously, they're bubbling CO2 and oxygen, mostly CO2 through it. They're exposing it to light and feeding it the nitrogen and the carbon and the phosphorus they need so that it produces oxygen and energy, right? Because some of what they produce is also hydrogen gas. And so we use it as a giant bioreactor. And in Hawaii, why not? It's sunny almost all the time. In Southern California, sunny almost all the time. Nevada does a lot of this kind of stuff because it's sunny 360 days a year. So yeah, cyanos are a really good example. Purple sulfur bacteria and green non-sulfur bacteria also work. So let's talk temperatures, all right? And so just very broadly, there's a minimum, a maximum, and an optimum. And how we define these is kind of interesting here. The minimum is the lowest temperature that a microbe will actually grow at, so that it will reproduce at. Below the minimums, and this is important, below the minimums, the cell goes dormant. It does not die. So if you were to put chicken that had been in your fridge a week in the freezer, right? And I know nobody would do this, right? I'll throw a couple winks in there. And you take it out a month later, right? You couldn't eat the chicken when you put it in. You should not eat the chicken when you take it out, because all of the microbes that were contaminating your chicken and making it smell bad when you packed it away will come back to life when the temperature comes up. So the minimum temperature inhibits growth. You may lose a few cells along the way, but by and large, your meat, your chicken, your samples, whatever you're putting in the freezer are still contaminated. The maximum makes sense, the highest temperature that the organism will grow, which usually means metabolically and reproductively. Above a maximum, the cell will die, right? Because you denature their proteins, their enzymes, and their membranes, and you render that cell inoperable. Endospores, in what way? So endospores, because they pull the water out, are dormant, and the maximum and the minimum would go away. So like a Clostridium is probably more of a mesophile. So if you grow Clostridium at above 40, it will die. If you grow it at, let's say, 5 degrees Celsius, it will be dormant. Once it makes the endospores, even if you heated them up to 100, or you dropped it to -80, nothing's going to happen to them until water osmosis back into the cells. Once that happens, once they return to their vegetative state, right, they go back to meeting the maximum and minimum requirement to divide. And since we're thinking about spores, the Clostridium spores would also require an atmosphere void of oxygen. So there's an additional requirement for that particular spore, while anthrax and some of the others would be removed from that requirement. Now, the optimum here is defined at the highest rate of reproduction. Right, so we're looking at the optimal rate of reproduction. And oftentimes for pathogens, that is going to be 37 degrees C. Now, that doesn't mean it's always 37. We're going to look at different temperature classes of different organisms, and they will all have their own minimum, maximum, and optimum. And some of these tests, like for E. coli, for example, E. coli grows very well at 42 Celsius, whereas a lot of things peak at 37. And so while E. coli would still have an optimum at 37 degrees, a test to identify E. coli might rely on a 42-degree maximum temperature to do that. When you get to 43, E. coli is done. It's not able to survive. And thinking about that 41, 42, 43 degree range, anybody quickly or just with a guesstimate know what that is in Fahrenheit? for lack of a better descriptive way, that is past the point at which your brain would cook from a fever. Right? 40 degrees is 104 Fahrenheit. So once you get to 42, 43, 44, doesn't do the E. coli any good to grow any faster or better because its host is gone. And I'm speaking human. If you're looking at cat, and I see a cat in one of the pictures, the cat's normal temperature is 102.5. I've seen cats get to 107 and live. So an E. coli might do a little better in those hosts, but in us, not so much. So just kind of experiential, that's some stuff to think about. Now, the organisms that we look at, right, all will have a minimum, a maximum, and an optimum, totally true, but we break them out into 3 temperature ranges. Now, our books and the lab book and the other book do have slightly different ranges. So on here, it says psychrophiles are between 0 and 15. Some books will go from -5 to 20. I think your books are doing this to eliminate the overlap so we don't have as much confusion. But a lot of the times, and I can even put this in here, -5C to 20C. Right, so it depends on where you look. The idea here, though, is that the optimum is typically around 5, typically, but results vary, and that they can grow at 0. So that means in the snow, right, if that snow is 32 degrees Fahrenheit or 0 degrees Celsius, these organisms could actually reproduce. What does that say about your refrigerators? Right, and these are things we don't like to think about, but your refrigerator is an incubator, is it not? And we say that the safe temperature is between 0 and 4 Celsius. So we want to keep it below 40 degrees Fahrenheit. That doesn't mean that nothing grows because your food spoils, your orange juice ferments, your pasta turns green. It just means that this stuff is growing very, very slowly because you've removed the energy. So I'm going to kind of, let's see, separately from this, there's something called a Q10.
This is why at a buffet, hot food should be hot, cold food needs to be cold.
And you know, even buffets, could you imagine, you know, now doing a buffet with COVID?
Blech.
But you have a good point there. You know, the salad dressings need to be kept cold. You definitely don't want somebody sneezing in it and having it reproduce to the point where you have staph or you have E. coli growing in your ranch, right? Obviously, we don't want that. And hopefully at buffets, you would find ice underneath those things. And, you know, the things that are supposed to be kept warm outside of that danger zone of 140 Fahrenheit to, I'm just trying to think of my numbers here, 60 degrees Fahrenheit, I guess. Anything that should be kept warm needs to be kept above that 140, otherwise bacteria could grow. And if the warming lights are off, you get a little worried, right? The carving station with the meat always has a big light while the guy's cutting. The pastas are always got a flame under it while she's stirring them. This way, we keep the food safe. And trust me, when inspectors come in, and they do come in, those temperature violations are red flag violations. Now, not to scare you, since you asked, I'm gonna volunteer. Taco Bell is one of the safest fast food restaurants you can go to. They don't actually cook on the premises. All they do is heat and warm. And if I told you they had just shy of 2 critical violations per 100 visits, would that shock you? Right, so they, you know, McDonald's is up like 98 critical violations per 100 visits, but Taco Bell is really low because all they have to do is make sure the hot tables are hot and the cold tables are cold. Yeah. And yet I can tell you that a kitchen in a restaurant is probably more dangerous than the buffet. And having worked in these kitchens, I'm sure there's some servers or some chefs in the room. Those kitchens hit 110 Fahrenheit. They hit 120 Fahrenheit in the summer. And so remind me again, a mesophile is 37 degrees, so it's 98, it would grow. That kitchen is easily 98 degrees. And so the cook, the counter people, the servers, I hate to say it, they have their headgear on, a bandana, something, because they're sweating. And that sweat drips places. hopefully not your food. And so you're inoculating or possibly inoculating food with sweaty hands, wiping your brow, and going back into these cold and hot things. Staphylococcal food poisoning happens as a result of that kind of environment, right? Staphylococcus is a mesophile. It grows between 20 and 40 Celsius. So the kitchen at 100 degrees is just below 40 C. It's not happy about it, but 6 hours on the steam table or 6 hours in the tray table there, it's going to double every 20 minutes at that temperature, right? So that's why foodborne illnesses and temperature go hand in hand. So our pathogens largely fall under the mesophile category. Yeah, sorry, I don't want to ruin it for folks, but you know, this is why the health this is why the health rules and the health departments are there the way they are. Because being, you know, warm-blooded, being our average at 37 C, mesophiles are where it's at. Your staph, your salmonella, your E. coli, E. aerogenes, so enterobacters are interesting. In delis and in pizzerias, sometimes folks have more than one job. They'll work outside, they'll come in and they'll cook in the evenings. And a lot of the places where these foodborne pathogens dwell is under the fingernails, right? Whether it's a restroom stop, whether it's a soil stop, whether it's trash, the stuff under the nails, fecal matter, you know, easily can be transferred to food. And at 40 C in a kitchen, it's not hard to see why accidents happen. So a mesophile, believe it or not, will grow at 20 degrees. That's about 68 Fahrenheit if we're counting. So that means, or this is why you try not to thaw your food on the counter. This is why you try not to leave food on the counter overnight. Because again, at 20 degrees, your E. coli and your staph are going to divide. At body temperature for a mesophile is where you see the optimum rate of reproduction. So for a mesophile, we usually say 37 degrees. I'm going to say body temperature because your body is not my body, is not someone else's. All right, so body temperature here is the optimum, usually defined as 37. And yet some will grow at 40. E. coli will grow at slightly higher, but really that range is about 20 degrees. Now thinking about the Q10 beneath, right? Metabolic rate measure sometimes gets a Q10 value, or we talk about Q10. So if something divides, let's just make up a number, every hour and a half at 20 degrees Celsius. At 30 degrees, it should double its division. So instead of every hour and a half, we should see what? Every, oh, let me do, I don't want to do the math tonight. I don't know, what is that, every 40 minutes roughly? It's probably not, but give or take. It's a little more than 40, a little less than 40. And if we make it to 40 C, then we should see it double every 20 to 30 minutes. So at 20 degrees, it'll be every hour and a half. But at 40 degrees or approaching 40 degrees, it might be every 20 to 30, 40 minutes. All right, and so that's the effect you have on temperature. Now, if there's E. coli in your steak, there will be. And you take it and you put it in the refrigerator, now you went below the minimum. And so it's going to take a lot more time for it to spoil because you've sucked all the energy out and you've destroyed, let me rephrase that, you've inhibited the enzyme systems. And so that steak is going to make it much longer time in your refrigerator than it would on your counter, right? And likely it's going to be something else that spoils your steak. So that's a good question. Organic food, organic vegetables, and things of that nature tend to have less pesticides and less stuff mixed on them. And so you find that they tend to rot more quickly. And that may not be necessarily a byproduct of the chemicals as much as the bacterial communities that are living on the fruit that were not killed by the chemicals. So if you look at strawberries are known to harbor fungi, right? And most of the times when you buy the strawberries at the supermarket, it's already on the fruit, it's already on your apples, it's already on them. And you take them home and you get a day or two before they're just fuzzy. There are strawberries that are grown with heavy pesticides, and there are even some that are irradiated that will last 2 weeks in the refrigerator without being moldy, right? And that's just a really weird thing. The other thing, if we're thinking organic, I'm also thinking non-GMO, non or less processed. They tend to spoil more rapidly because there's not added sugar to it. There's not added salt to it. and there's not added preservatives to it, all of which are meant to inhibit bacterial growth. The addition of sugar, right, has been known back to Napoleonic times to be a way to keep wounds from festering. So a lot of that food does spoil faster. However, the benefit to organic is usually it gets to the market faster. So there's still no substitute for fresh, if that makes anybody feel better. Or maybe it doesn't, but either way. Let's see. Oh, and the last ones are our thermophiles. So the optimum temperature, and again, this is going to vary depending upon your book. Generally, you'll see 45 degrees C. You might even see greater than 60 C, depending on the book that you see. Within the thermophiles, though, there are a couple deviations we should talk about. One of them are facultative thermophiles. One of them are obligate thermophiles. So I'm hoping that you guys talked about facultative anaerobes and obligate anaerobes when you talked about oxygen requirements, but if not, no big deal. Facultative means can grow less than 60, but has an optimum of greater than 60. And obligate means it must grow more than 60. They're close enough. I would say, whatever the book is telling you is what we're going to go with. I'll say that on video for us. So whatever the book says is what we're going to do. But I also don't want to mislead folks into thinking that one author's interpretation is the be all and end all. I'm sure Tolero and I'm sure the Pearson book differ in this area. So in fairness, maybe we just say we go on what the PowerPoint originally said. So this in parentheses stuff you can ignore, but I want you to have that information anyway. Biology is the land of exceptions, and the temperature rules are not an exception. So facultative thermophiles will grow at 37. It'll grow at 38. It'll grow at 40. Obligate thermophiles grow only at 60 or more. And so thermophiles don't make good pathogens, as one of the places we're going to go with this. And if you keep watch, there are also extremophiles that will grow much greater than 60 C. And 60 C gets us to what, 140 Fahrenheit? Does that sound about right? I think that, well, close enough. So not the hottest place on Earth. There's places that are certainly warmer. But there are places like hydrothermal vents or hot springs or even Old Faithful up in Yellowstone that'll reach 100 degrees C, 110 degrees C, 120 degrees C. And the hottest organism that I've ever seen grow at 123 C survived over an hour. I just want to point out that at 120 degrees C, water has long since been boiling. So you can imagine the kind of organism that grows here and the amount of energy that it actually uses to reproduce and metabolize, right? 120 C, pretty amazing. I guess I'll ask one other question. Do folks ever make their own fermented stuffs at home? Have you guys ever made your own, I don't know, Applejack or pickles or kimchi or sauerkraut or anything like that? Just kind of looking at the chat, looking here. Never, yeah. Honestly, you shouldn't be afraid of it. And it's honestly not really hard. If you're into the whole microbiome thing, there's supposedly, the stuff is very good for you. And what you do is you make the environment in the, oh, there we go, my parents. So they got some wisdom that they can drop on us. What you do is you make the environment in these jars only hospitable to organisms that would ferment correctly. And that sounds really weird. But in the case of pickles, it's Acetobacter. So you add acid, you add a bunch of salt and a bunch of spices, and you leave it open. You actually just leave it open to the air, hopefully at less than 20 degrees Celsius. And the organisms from the air inoculate it and start to do the biochemical transformation from cucumber to pickle. My wife always calls them rotten cucumbers, and I think she does it to piss me off. And I can't even get her to understand, look, it's not rotten. It's just changed. It's fermented. It's different. It's like a beer or wine. And honestly, pickles are great. I could eat them all day. So it's one of the things I kind of play with. Kimchi is awesome, especially when you get the recipe you just like. But I used to make Applejack as a kid. I used to go up to the Conklin Orchards up in on 45 in Pomona. I would get the gallon or two, depending on when and how much I wanted to make. And I would buy some of their apples. And the apples, like most fruit, have a wild yeast on the skin, right? So it's their normal flora. And I would skin them, I would throw them into the jars, close them up, put them in a brown bag, and set them in the basement, which was about 55 degrees. And 3 weeks later, at that temperature, I would have apple cider. It might be a little moldy. I would have to filter it, get the apple skins out. The mold came from the cores, from the skins, and I would filter it out, and I would have roughly a 3% alcoholic beverage. It was actually pretty good. Now, in your refrigerators, I guarantee you someone's got a bottle of orange juice or a carton of orange juice from the holidays, right? And I mean like Christmas, New Year's. Someone's got to have one. And that's a good 3 months, not a good 3 weeks at this point. We're coming up on 3 months. So I guarantee you, if you're one of those people, go in there and open it. And you're going to hear gas release, right? Evidence of fermentation. Give it a sniff. Might smell a little sour, but you'll know it's alcoholic when you take that sip. Right? If there's a little mold on it, no problem. Just skim it off. It's not a big deal. And people are making faces. All right, so let me ruin something else. Pizza sauce is one of those things that I know you love or you hate, but you go to a place because it has the right sauce. The pizza sauce is refrigerated when it's not in use, and we usually take some out and have a working container. This way, when that's over with, we don't contaminate the stock. Would you believe that pizza sauce in that refrigerator, 5 gallons or more of it, tends to get moldy, especially if it's been a slow period? And what most restaurants do is just skim the mold off. Now, hopefully your pizza shop is busy because you like it and you go there. But, you know, in some of these rural areas, like who eats Vermont pizza, right? Anyway, I don't want to get into an argument. You can use it, though, until it starts to ferment. And once the pizza sauce ferments, you got to throw it away, right? So there's a lot of this in food microbiology that, unfortunately, we're not looking at because, well, we're interested in humans, right? But it's kind of wild when you think about it. Now, graphically, this is kind of what you're looking at, and there's some overlap here. And so I do want to address the overlap. So if you have an organism that grows at 15, right, notice there's an overlap between the psychrophiles and the mesophiles. You guys being the good microbiologists, how would you figure out if you had a psychrophile or a mesophile? What would you try and do? Because they're both going to grow at 15. What do you do? Right, so people ask like test questions and how could I study? This is an application of what we just talked about. It's not asking you what a psychrophile is. It's not asking you what a mesophile is, but it's asking you to apply what you know about psychrophiles and mesophiles. So it's a good question, but it's not a good question, right? Because it's not the ones that folks want to see. So thinking about 15, right? There's an overlap. The blue line touches it. The green line comes over it. What do we do?
Do you heat it up a little more or cool it down a little?
Right, so try growing it at 5 or 0, and try growing it at 37. Wherever it grows will be the differential test. If it grows at 0, it's a psychrophile. If you don't see any growth at 37, you confirm it. If you see growth at 37, it's a mesophile. You will not see growth at 5 if it's that case. So it's a good way to discern or to test. The ones that get a little crazy is if you were to take a thermophile and grow it at 37 versus taking a thermophile and growing it at 60. If it's a mesophile, you will see abundant growth. If it's a thermophile and it is facultative, you'll see scant growth, but you'll see more abundant growth at 60. So you probably, yeah, you do have to expose it to different temperatures, and you've got to just do a quick experiment. So now that I've ruined a bunch of things, refrigerators, pizza shops, how do we feel about temperature? So I'm just going to hit stop here. And I know that was one of the lab components. Do you feel better off or worse off? I don't mean about your kitchens or your food, but about the temperatures now than we did before. Yes. I leave it on the counter all the time. It's going to be eaten until it's gone. But the health department would be very upset with you if you left like a pepperoni or a sausage pizza on the counter. What you're supposed to do is have a display slice. And I'm not kidding, a display slice. And then the stuff you sell is underneath the counter in the refrigerator. I wonder if you think pizza would be ruined if you put like anchovies on it or cucumbers, right? Does that ruin pizza? Oh, see, we have a purist in the room. That's fine. There was a really good Seinfeld episode about Kramer putting cucumbers on there, and it drove, I think his name was Poppy, crazy. We can mess pizza up. We don't want to, but we can. All right, temperatures seem to be good. All right, we're solving problems tonight. This is a good thing. Let me pop over here. All right, so temperatures we're going to kind of pop over through. And I'm going to click on this one next. Now, I know this was part of the lab. And this is an interesting one. And by interesting one, this is probably the experiment that I love doing the most. Because in doing this, well, let me walk you through it. So what we do here is we use an agar deep. And so that means we make a nutrient solution, or at least a solution that we know is going to feed the organism, taking into account any nutritional requirements we need. So if this were a strep, we would use blood. If this was a fungus, we'd use sabarod. But generally, we're going to use nutrient agar to do this with about 1.5% of agar added to it. So we fill it up about yay. We autoclave it, and it comes out hot, and then we let it cool till we're ready to use it. The night that you guys would do this, or the day that we wanted to do the experiment, what we would have to do is reliquefy the agar. And so we would probably boil it in water, or at least microwave it, and that's kind of cheating, but it works. Microwave it to get it to liquefy. And you need at least 60 degrees in temperature to do that. You would then keep these in a warm bath at 50 C or higher until you were ready to inoculate. Now, the details of how that happens vary, but essentially someone's going to open the tube. One of the other group members is going to take a loopful or a drop of microorganism, put it into this agar deep, which is now liquefied, and you're going to take it in your hand. Let's see, got something in my hand, right? That's our test tube. It's just a fat Sharpie. And you would do this with the Sharpie, the test tube filled with organism and liquid agar that you just inoculated. And you would do this to fully disperse the microorganism in this liquid agar tube. Someone else from the group in a rack would bring it up and put the rack into ice water. And the ice water will cause the agar to solidify immediately. And so the school of thought here is it's liquid, you inoculate, you fully disperse by mixing it, and then you freeze it where it went. And so theoretically, you should have organisms spread throughout your tube. Then you incubate them based on how you're supposed to. So if these are aerobes, well, if you're not sure, you just put them in the incubator and you let it go. If you need to put carbon dioxide in, fine. If you need pressure, fine. And then based on where it grows in the tube, we start to draw some inferences. All right, so this is where things get a little challenging. Remember, we inoculated the whole tube, right? So this whole tube has bacteria or your culture in it. And what we're seeing is where it grows. And where it grows is indicative of its oxygen requirement, okay? So an aerobe requires oxygen. And so an aerobe is going to grow in the tube wherever it has enough oxygen to meet its metabolic demand. And so we would think largely that would be in the top quarter to top third of a tube. And so an aerobe like, I don't know, E. coli would follow that pattern. Now a microaerophile, right, tolerates oxygen. Right? It needs just a teeny tiny amount. And so full strength oxygen is toxic, right? It would oxidize the cells and cause damage. So it lives in the oxygenic region, but below the surface where the oxygen is much less in concentration. And so even though we inoculated the whole tube, you're going to see growth just beneath the surface here in a very discrete band. The example that we normally give here are things like yeasts. In the lab, the example we would use is Saccharomyces cerevisiae. Right? So it's brewer's yeast. And essentially, it tolerates a small amount of oxygen. And think about when you add this to a wine bottle or a grape juice bottle or an orange juice bottle or an apple juice bottle. You put the lid on and you seal it up. So whatever oxygen is in there, and the fermentation starts from deep inside the juice, not near the top. Eventually, there's enough CO2 in there that it will eventually ferment the entire product. But a microaerophile grows just under the surface where oxygen concentrations tend to be a little less. I'm going to skip the middle tube briefly. I'm going to come over here to the aerotolerant anaerobe. All All right, and what you're seeing here is it's growing where there's still oxygen, but it prefers to be in the lowest amount of oxygen. So basically from mid-tube to the bottom, where the concentrations of oxygen would be lowest because the rate of diffusion would be lowest. Now, a microaerophile, and I don't know if you talked about this in the lab, so if you didn't, stop me. Microaerophiles have the enzymes to deal with oxygen and superoxides. And what I'm getting at here is superoxide dismutase and catalase. And if those are foreign words, maybe I'll put those in the chat. That might have been spelled wrong, but it's superoxide dismutase, that I spelled right, and catalase. I don't know if that Y is in the right place, but I'm not trying to spell check that now. Those are what organisms use to deal with oxygen and superoxide radicals. So what ends up happening is the oxygen with superoxide dismutase is turned into hydrogen peroxide and water. And then the catalase takes the hydrogen peroxide and it turns it into water and CO2. No, sorry, just water and perhaps some free regular oxygen, not superoxides. So the oxide dismutase turns it into catalase and water turns it into hydrogen peroxide and water. And then the hydrogen peroxide is turned into water with some oxygen gas released. But it's oxygen gas, not superoxides and not super radicals. Did you guys talk about that? I'm not seeing a lot of yeses. So while you guys are pondering that question, let me just find something for you real quick. I would assume this would have happened in the lab, but maybe it didn't. So these super radicals, basically, we would take these super radicals on the left. Sometimes it's an HO2, sometimes it's just an O2 minus 2. And it turns them into less harmless oxygen and water. And then catalase, oh, so there isn't a Y in there. That's my bad. And then the catalase does this. So it takes the hydrogen peroxide and it turns it into regular water and oxygen. So these two enzymes allow microaerophiles to deal with the oxygen. It allows aerobes to deal with the oxygen. And they're present in aerobes and facultative anaerobes, but these enzymes are not present in aerotolerant anaerobes or anaerobes. And so they lack the mechanisms to deal with oxygen. So the aerotolerant anaerobes have to avoid it. The anaerobic organisms cannot be in the presence of oxygen. And so that's why in this picture here, they grow at the very, very, very bottom. Now, in the lab, we use, let's see, probably E. coli, probably Saccharomyces cerevisiae for the microaerophile. Does anybody remember what you used for the facultative anaerobe? And I'm sure you don't, and you don't have to go back. I'll give you a little blast in the chat here. Staphylococcus aureus is a facultative anaerobe. Now, I don't know if folks have been eating dinner or not. Maybe you have, maybe you haven't. I'm going to find something else for us. Let's see. Let's see, maybe. Maybe, let me check this one real quick. Turn off the sound. Good, fine, keep going. Oh, this one was boring. Trying to find a good video that illustrates this point. I don't know why that matters. All right, not a good example of that video. No, maybe. This is good. All right. All right, so I'm going to switch over. So Staphylococcus aureus is a really good facultative anaerobe. And what that means is it has the enzymatic machinery to deal with oxygen. So it's going to have the superoxide dismutase and the catalase. When the oxygen is used up, it has the ability to switch from aerobic to anaerobic pathways. And the importance there is really, it's an extra tool for Staph aureus to make us sick. And so while it has exoenzymes that give off toxins, it also has the ability to work with and without oxygen. Now, I don't know how your stomachs are. I don't know who's eating dinner, so we're just going to call this a heads up. Some of the things or some of the mechanisms that illustrate this really well are sebaceous cysts. Right, and I always tell the story, or maybe you've heard the story. When I was 5, my dad and my grandfather both are sufferers of sebaceous cysts. And unfortunately, I inherited that trait. And so 5-year-old me went with 40-year-old dad to the doctor's office and watched them, not because I wanted to, but because my mother was at a doctor's appointment, watched them lance my dad's boil, or his sebaceous cyst. It was the size of a cantaloupe coming out of his back, which meant it was also the size of a cantaloupe going into his back. And in the center of these sebaceous cysts, they are typically anaerobic. All right, and so what typically happens when we pop our own stuffs is that you get a white core of staph out, right? So you get a core of microbe out. Sometimes it's hard, sometimes it's a little runnier and greener, depending on how it's been infected. Yeah, exactly correct. Except I had a front seat. And the smell is something that you can't describe when you watch Dr. Pimple Popper. So needless to say, I got through the first couple layers of redox. We went from white to yellow, we went from yellow to green. Once we hit the purple, I had enough. And I started to turn green myself at 5-year-old, and they took me out of it. And then the real fun was keeping it open, because if you let it close, then the anaerobic conditions come back and the staph multiplies and the cyst continues to grow. So just to give you an idea of what that means or what that looks like, and I don't mean to be gross, and I'm going to fast forward some of this because obviously you don't want to see this. However, if you have a weak stomach, if you just ate dinner, maybe you want to fast forward this or like not look for a little while, because it might be a little skeevy. And I don't know how much sound is in here. I don't even know if it matters, but the first thing they're doing is injecting a lidocaine and antibiotic mixture. I can tell you this because I've had them in my armpit, and that's the first part. Then they're going to open it. Now, sometimes the antibiotic and the added pressure will cause it to pop. What they're actually opening is the cyst. And so typically the cyst is fibrous. And so you're going to have to open it and remove it if you don't want it to come back. It's not always possible to do it at the same time. So they're cutting into this person's skin, absolutely, but they want to make sure that they puncture and get into the cyst. Oh, and there it goes. So I don't know if you saw the spray, but that was the stuff they just injected. And yep, there it goes. And it's still not quite done. And now you're seeing the staph, right? So that's Staph aureus. And notice it's getting greener and greener. And there's a little purple coming out, and there's a little bit of blood coming out. And so once it turns, a little orangey, although that's a little blood. Once it turns from yellow to green, you've gone from aerobic to anaerobic. All right, and this is just ridiculous, the amount of fluid that's in there and the amount of fluid that they're pulling out. Sometimes it's runny like this, sometimes it's a little more like cottage cheese, but what you're really looking for is that color change. All right, and so they're really gonna get in there, and you can see, and oh, there was a chunk. That was a piece of purple tissue. And they're gonna just keep going here and make sure they get it all. Oh, there's some more. Now, I don't know what this doctor's gonna do. I don't seek these videos out. Oh, she's got her finger in there. That's nice. They're gonna try to pull out. Yep, there you go. They're gonna try and pull out the fibrous portion of that. So they're gonna try and get out the actual lining of the cyst so that this person doesn't have to get this drained again. Right? And I don't know how. Oh, there we go. That's the rest of it. Yep, she's doing a good job. Some gauze probably was in there. Yep, she's trying to get it all. We probably don't need to see all of this. We probably don't need to watch her suturing all of this. All right, but the point here was when it went from white to green or yellow to green, so somewhere around this part, right, where it still looks whitish, and then you see wherever that's coming from, it's got that green tinge to it. That's how you know you've hit the redox boundary. Oh yeah, there we go. You've gone from oxygenic to non-oxygenic, or it's aerobic versus it turned facultative anaerobic. So yeah, we probably don't want to look at that anymore. We'll kind of stop that there. Little graphic, sorry about that. If you guys were doing one of these, you can look again. Yeah, it is kind of gross. And yeah, it is kind of ew. And the smell doesn't do it justice. But that's what staph does. Staph, if it didn't switch from aerobic to anaerobic, no, I'm not sorry, you're right, would have never gotten that deep. It just wouldn't have happened. It would not have been able to survive without oxygen. But the fact that it able to switch allowed that thing to be a monster. And so that's just one of the extra goodies that Staph aureus has that allows it to do the things it does to us. And it's kind of mean, it's kind of nasty. But anyway, I'm sure you're going to see much worse on the job. So we might as well take a quick look now while we're thinking about it. Let's see. Not that screen, this screen. The last one here, we did not have a tube that looks like this because we would have to grow this without oxygen in order for you to see really good growth. And so what we tend to do is instead of using a tube like this, we tend to use anaerobic chambers. And down here in the bottom picture is what an anaerobic chamber typically looks like. And so I think in the lab, we've resorted to Tupperware and gas packs. In some of the higher tech labs or the labs with a couple more bucks, what you have is a glass or a plastic lid where you put your tubes and your plates, and the lid has some catalyst in it. Now, in this particular one, it's palladium, and some of them it's platinum. And what ends up happening is the gas pack produces hydrogen gas and CO2. The hydrogen reacts with the oxygen to form water. So it essentially converts the atmosphere from one of oxygen to one void of oxygen. And if anything in here is anaerobic, it will grow, provided it's warm enough and it's got enough pressure and it's got the right pH to become very numerous. And so typically the lab organism we use for this purpose is Clostridium. Now, it's not Clostridium difficile, so it's not C. diff, the one that you're used to seeing or smelling. And if you've ever smelled it, you never forget the smell. It smells kind of like a diaper genie. I lied. Well, it's a sweet diaper genie, I guess I would want to call it, because it also has like a grape sugary smell to it. Clostridium sporogenes is the one that we typically use in the lab. That one smells like rotten diaper genie. There's no sweetness about it. And what you're smelling, those herbs and spices, are just the byproducts of metabolism, right? So it's not meaning to offend, it's what it's producing. And so we would typically put the things in, add the gas pack, add a little water to the gas pack to activate it, and then let her rip. Put the screw on, slide the whole thing into the incubator, and see 24 to 48 hours later what grows. Now, on the other ones, I'm going to tell you it's 12 to 24. Why do you think I need 24 to 48 hours to see an anaerobe grow, or at least an obligate anaerobe in this case? Would there be a reason or a purpose that I'd have to let it incubate longer? I don't think this would be a test question, but it's a good question nonetheless. So this has to do with energetics. Oxygen and oxygenic final electron acceptor reactions produce on the order of 700 kilocalories per mole of glucose using oxygen. When we get rid of oxygen, we drop at best, when we see nitrate as the final acceptor, to a little under 500 kilocalories per mole of glucose. If we go down to the worst, like CO2 or methane or sulfate, we're down under 100 kilocalories of energy available per mole of glucose. So with the removal of oxygen and the using of other electron acceptors to complete their metabolism, you've removed a great energy source. And so in order to see growth, you're going to have to let it go a little longer, because it's just not going to double as quickly as it would with oxygen present, or an organism that has the ability to use oxygen. And that would go for staph as well. Staph with oxygen versus Staph aureus without oxygen, you would see a lower rate of reproduction. There just isn't enough energy there to do it. So I'm going to just quickly go back here, if it'll let me. Yep. and just ask, okay, I know you guys did this in the lab. You didn't have the opportunity to do the experiment, which is unfortunate because it's a fun one. Does it jive with the stuff that you guys talked about in the lab in terms of what the results mean and why? Right? Or if you didn't talk about it in the lab and you have it next week when you see the results, awesome. Then you're going to have that leg up, and it should be a review and or boring. I don't know which way it's going to go, but hopefully that was helpful. And you'll have the video to play back just in case you want to look at it again. All right. I just love it in the chat. You're not sorry. Nope, I'm not. All right. What else is in here that we want to talk about? Yeah, I suppose. All right. So there's the usual relationships that we might talk about in here, symbioses, if you will, organisms living in close relationships required by one or the other, right? So it's not necessarily both members, but it's one or the other. Maybe it's both. And so mutualism, commensalism, and parasitism are the three that we tend to look at. Right? So mutualism is when both members benefit. So everybody's getting something out of the relationship. I live in you, you provide me with nutrients, I provide you with vitamins, right? That would be a really good example. We have commensalism, where the commensal member benefits, others are not harmed. So there's probably organisms in our gut that are there that aren't really giving anything back, but they're not really harming us either. And I really hate to use that as an example because just by virtue of them being there, they're giving something back. They're taking up space for something else nasty to move in and make us sick. So maybe not the best example, but they're basically getting something out of it. We're not being harmed. That's really more to the point. Whereas with parasitism, the host is harmed. We're harmed inadvertently, I will add, while the parasite gets larger, reproduces, and makes more of itself. You got to think of it this way: the parasite doesn't want to kill us. If it kills us, it's meal ticket. But unfortunately, you can only coexist with some things for so long. One of the more interesting things, or interesting ones, we think about is usually when animals or worms get inside us, right? We think of them as parasites. They're doing us harm. They're stealing our nutrition. And it's kind of interesting because you're saying, well, they don't really hurt us. They're just taking a little bit, but they are, right? Because they're taking the food that you need to do your metabolism with. They might be making harmful byproducts. Rickettsia probably would be, because it needs a host in order to survive, and it can't reproduce without the host. So yeah, I would consider Rickettsia and even the Lyme disease spirochete to be parasitic, irrespective of them being gram-negative spirochetes or them being strange organisms without cell walls, right? Things of that nature. The classic example, though, that we think about are tapeworms. And I guess it was common at the turn of last century, so the early 1900s, for people who were obese or who needed to lose weight or who thought they needed to lose weight to eat tapeworms. So to purposefully consume them so that they would lose weight. Now, I've heard of examples where on death, the tapeworm will crawl into the esophagus, will crawl out of the anus looking for food, and there's been some interesting autopsies on that. But normally you would go to the physician and they would give you a pill, and the pill they give you would be to dissolve the scolex. And then you would pass the worm out, right? Getting rid of the nutrition sucker from you. And sometimes these worms are meters in length. So one of our colleagues at Westchester, and I won't tell you who it is just in case you know them, but she'll tell the story if you ask. Her husband had a tapeworm. And when he was passing it, don't ask me why, this is 1970s, she went in there with a pencil and took the worm that he was passing and wound it around the pencil so that, I guess she was helping him pass it. In hindsight, not a good idea. You don't want to pull on a tapeworm because you could break off the proglottids, the sections of mature worm eggs that could produce more tapeworms. And she put it in a jar. Right? She put this worm from her husband in a jar so that she could bring it to school and show students. So as gross and terrible as it sounds, the motivation was really there. Yeah, I know, scientists do sick things. That's not the only thing we put in jars, by the way. I'm not going to go there. But there are individuals who have had these tapeworms, rather than going to the doctor, they decide they want to keep them, right? They want to hang out with them. And so there was a guy who had a tapeworm, and it burst through the abdominal wall. It actually was on the outside of his abdomen. And he called it his pet, and he would feed it bread. And you know, being a parasite, it's bent on getting more, and more, growing and growing. It also burst into his inside, and it started to consume his abdominal organs. Yeah, Google it. You can find these things on the internet. I'm not sure you'll want to find it. Just Google this stuff. The information about the lady in the tapeworm, she's since retired. So maybe I can spill the beans. Her name was Iris Cook at Westchester, Professor Cook. So you could ask the questions, you could find things out. I just want you to ask the questions that you only want the answers to, because sometimes you don't want these answers. Sometimes we sometimes we have organisms, and we've talked about the idea that sometimes we can't grow things in a pure culture. And so the organisms are not living inside one another, but they're living as a community. And in a community of dwellers, whether it's humans or cats or dogs or giraffes or what, doesn't matter, there's organisms that cooperate and work together, right? So you see synergism. And then there's members that fight with each other and try to destroy one another, and that's antagonism. And so with bacteria, we often think of complementation as a synergism. I make lysine, you don't. You make histidine, I can't. So if we live together, you give me what I need, I give you what you need, and we fulfill this niche in an ecosystem. And so it's a little different than symbiotic relationship, because in symbioses, the organism is within the other. Here, they're free living and they work together in the environmental realm. So it's kind of an interesting way to look at things. And I suppose, we don't talk about it here, but there's something called endosymbiotic theory. And I don't know if anybody's heard of that necessarily. And it's the theory by which prokaryotes became eukaryotes, right? So a prokaryote sees a free-swimming, a free-roaming mitochondria, and it engulfed it. And rather than engulfing it and hydrolyzing it, it allowed it to stay. Or maybe it couldn't hydrolyze it, I don't know. We weren't there. And so rather than destroy it, it let it stay, and it learned that it produced large amounts of energy using oxygen. And so when that cell divided, it divided with the mitochondria, because the mitochondria have their own DNA and are self-replicating. And so now this original eukaryote, by way of engulfing a mitochondria, prokaryote became a eukaryote. And likewise, because mitochondria and chloroplast are very similar, a free-living non-photosynthetic eukaryote happened to see a chloroplast. It engulfed it, but didn't hydrolyze it, did not kill it. And it learned quickly that it produced sugars and food. And so it left it alone. And when that cell started to replicate, right, thinking of the dawn of plants, it replicated not only the mitochondria and the other organelles, but the chloroplast as well, because the chloroplast has its own DNA also. So it's different than the symbioses that we've just talked about by virtue of, this is ancient history. We don't know for sure that that's how it happened, but it's a proposed mechanism to explain how prokaryotes became eukaryotes and how eukaryotes became plants and photosynthetic, right, photoautotrophs. It's an interesting theory, but it's a little outside of the realm of these two, but it's a third piece of the puzzle that you guys should probably see. All right, how we doing? We doing all right so far? Not too fried. It's not terrible. Moving along. All right, microorganisms and how they double. So if I told you mitosis without a nucleus, you would probably say, oh, that makes a lot of sense. What if I told you this is probably where mitosis came from? You'd say, that probably makes a lot of sense too. So basically, you've got an E. coli, well, we can call it an E. coli, and you're witnessing the E. coli growing larger, duplicating its nuclear contents and cytoplasmic contents, growing a new plasma membrane and cell wall, separating and becoming two. So the idea of binary, one, fission, splitting apart. One cell grows and splits apart into two. And it's growing and becoming larger very much for the same ways that our cells do. They grow, they become more replete, they become more flush with nutrients, they become more advanced, And we hit that volume to surface area relationship boundary, where the inside is greater than its touch with nature. The inside has lost the ability to move things adequately across the membrane and bring in anew. So CO2 builds up, toxins build up, oxygen concentrations are low. And our cells have the same choice, die or divide. And so what happens here is the nucleoid region, right, chromosome where chromosome 1 is, undergoes DNA synthesis as the cell grows around it. And the DNA synthesis makes an exact copy of the chromosome. And while that's happening, it makes a copy of the cytoplasmic contents. This being a prokaryote, what could those cytoplasmic contents be? So cytoplasm should be somebody's response. Chromosome, obviously, might as well say that. Ribosomes, yeah, that would be fair. Ah, that's the one I was hoping somebody would say. And cellular contents also mean plasmids. So if you have an organism that's antibiotic resistant, or that has recently become antibiotic resistant, or even just one that's been transformed, like it could very well be from a pilus, and it could very well be that it's just naturally competent. It saw foreign DNA and it took it in. In the case of Streptococcus pneumoniae, that would happen that way, because Strep pneumoniae is gram-positive, no pili there. All right, so the plasmids are also replicated if they have any. And so that's how one single antibiotic resistant, metal resistant, chemical resistant cell can become numerous really, really fast. And so the cell membrane grows between the two cells. The cell wall grows in between the two cells, and they finally separate. Now, on average, if you're talking about an E. coli with good nutrition, this can happen every 20 to 40 minutes at 37 degrees Celsius. It might happen every hour to 2 on your counter. That's the typical rate for a pathogen. That's the typical rate for something that lives inside us. There are things in the world that double once a day under some of the worst conditions and under some of the best conditions. And so pathogens do have that advantage by virtue of metabolism, enzyme systems, their genetics, and the hosts and the environments they live in. Not all organisms double every 20 to 40 minutes, and that should be a reassuring thing. Unfortunately, the ones that make us sick do have that ability. So, okay, 20 to 40 minutes sounds scary, but again, 2% of everything is pathogenic, so largely we're not concerned. When these cells start to grow, all right, and I'm going to show this quickly before I look more at the S-shaped growth curve. There's a lag phase, there's an exponential phase, and then there's a stationary phase of cell growth. So E. coli, a microbe, whatever. And so we see 1 to 2, 2 to 4, 4 to 8, 8 to 16, the way we would expect through binary fission, the way the cell would grow. And the lag phase here is either the exposure to a replete environment, the exposure to an environment with nutrients, or getting the nutrients into the cell. So the smaller the organism, the less or the lesser of the lag phase. A bacterium might see a couple hours. A protista might see a couple days. Complicated protista might see 2 weeks. Small animals, 2 weeks, as we get into from stationary to log. And notice as time goes, a population that starts very, very small gets very, very large very, very quickly. So, you know, if we consider every 20 minutes, somebody keep track, right? 1 to 2, 2 to 4, 4 to 8, 8 to 16, to 32 to 64, to 128 to, so 128 is 10 divisions already, by the way. I'm sorry, we'll keep going. 128, 256, 512, 1024. So from 1 cell to 1024 is 10 divisions. 1 cell to 1024 cells, 1024 cells is 1 division, is 10 divisions. 1024, 248, right, 2048. And you can see how quickly the numbers go from there. Right, so this is why we call it log phase or exponential phase. And the numbers go from what, 500 to 4,500 in a short amount of time, as you see from this curve. Now, typically, this is the classic S-shaped growth curve. right? So we go from like a cell to, and I don't know, it's 5 hours, okay, to like, I'll even round, to 10 to the 2 cells. So we're at 100 cells. So in 5 hours, we go from 1 to 100. And then in 10 hours, you're at 10 to the 4 cells. So we're getting at what, 10,000 cells from 1 in 10 hours? At 15 hours, we're at 1 billion cells. So in increments of 5, we go from 1 to 100 to 10,000 to 1 billion. 1 billion cells per mil. That's outrageous. That's insanity. That is crazy. That's how quickly binary fission and how effective it is. So if this is food, right, you can see how it would spoil in 8 hours. If this was a blood infection, you could see how it would get out of control within 24 hours. Somebody might die. Now, in looking at this, we call it an S-shaped curve because of that S shape, right? The lag to the exponential to the stationary phase. So it's got this kind of S shape to it. Exponential is just that. You have extremely high cell division and extremely low cell death. So the numbers are increasing very rapidly. Then at some point, the nutrition in the organism, or the host dies, is used up. And the cells start dying at the same rate at which they're reproducing. That's why it's called stationary. We're not increasing, but we're really not decreasing the number of cells in culture. And so the cells die, they're replaced. They die, they're replaced. Eventually, if you extrapolate out long enough, you start to see cells die. And they're dying because of a buildup of nutrients like CO2 that shouldn't be there, or a lack of carbon, oxygen, nitrogen, and hydrogen, right? They're not able to build more cells. Now, as the cells die, the numbers will decrease. And at the very, very end, you see the dot, dot, dots, right? I love the dot, dot, dots here. What will begin to happen is the cells that die lyse. And so sometimes you'll see an oscillation of death phase, lag phase, death phase, lag phase at the end. Log phase, death log, where you'll see a little bit of an oscillation, or you'll see kind of a roller coaster at the end. The cells die and lyse, they're releasing their carbon, nitrogen, oxygen, and hydrogen back into the medium. It's called remineralization. And so the cells that are still alive can use it and then divide, but then they use it up again. And so we go back to a death phase. And then enough die and enough nitrogen and carbon and hydrogen is available again, and they start to divide again. So this could happen multiple times over the course of an older culture. What we try to do in the lab, and what you would try to do in a hospital or a research setting, is to use the cells in the log phase or the exponential phase. And you use them because you have the greatest number of viable cells. They are the strongest cells. They are going to react in the best possible manner. And it gives you the best possible idea of how fit are they and what are they actually doing. So the lag phase is just getting going. The exponential phase is the useful phase. And then stationary phase might be where we store them in the refrigerator ad nauseum. Remember, if this is E. coli, I can keep it at 20 degrees for a fairly long time before I have to transfer it again. Right? And so when I say transfer it, I mean I would get a brand new tube with brand new nutrients that I've never grown anything in, inoculate it the way you guys would in the lab, so a couple drops or a loopful, and I would start the process all over again. Those are the organisms that I would give to you in the lab. I would never take an old culture out and give you stationary phase stuff. Why? Because I don't think it would work. At least it would not work every time the way we want you to see it. Right? Whenever I do plasmid studies, whenever I would grow cultures that I wanted to freeze or archive, right, put them at -80, it would always be an exponential curve organism that I would use because they're the strongest and they would end up being the most viable. Now, the other thing I suppose I should tell you is, obviously this is happening in people, right? And so somebody might be inoculated and there's a lag phase. So you step on a nail, you stick yourself with a staple, right? The infection doesn't get going. You really don't notice it until the exponential phase, right? Maybe the best example is, I'm sure someone's had their ears pierced that remembers, right? And maybe it didn't go quite so well. Maybe there was an oopsie, right? You go home and your ear hurts. Yeah, they just put a post in it. And then maybe as you're cleaning it later on, you start to see discharge, right? It's probably on the order of about 12 to 15 hours, right? It fits our curve pretty well. So if you think of it from that perspective, it makes a lot of sense. Makes a lot of sense how somebody can go from okay to in danger very quickly. And I don't want to belabor that. But I will stop and I'll ask if there's questions and stuff like that. A little bit of philosophical, a little bit of theoretical, but it's good stuff. It's good stuff, no? All right. I think one of the other experiments you guys were working on was the use of, come on, a spectrophotometer. So this one expressly was about nutrients that you guys were using in the lab. And so you had 4 test tubes. I hope you saw this. I hope this is what you guys still did. A red cap, a green cap, a yellow cap, and a blue cap. And in order of red, green, yellow, blue, it should have been inorganic salts, glucose broth, nutrient broth, and yeast extract broth. And you inoculated four organisms, it's either 3 or 4, into various red, green, yellow, or blue tubes, right? So from something that was not fastidious to something that was very fastidious. And you use this spectrophotometer, which shines light through the sample, and it measures either how much light was absorbed or how much light was transmitted, depending upon how you have it set. So you're thinking to yourself, well, all right, I guess. Why would light be absorbed or transmitted? Well, cells tend to block or scatter light. And so the denser the number of cells, the cloudier it will be. The cloudier it will be, the less light should be transmitted or the more light that should be absorbed, right? Sometimes with fastidious organisms, you end up with scant growth. So very light growth that your eyes may not be able to detect. Now that doesn't mean nothing happened. It just means you don't have the tool to see what happened. And so your eyeball fails you, but the machine is more sensitive. And so you take a sterile tube, you wipe it clean as your blank, you read it, you set it to 0, and then you put the other tube in, your sample, and you see what percentage is transmitted or absorbed. Right? And then there's a correlation based on the absorption and how many cells. We never do the math, we just record the value. So next time you should see the least amount of growth in your red cap tubes, more growth in the green, more growth in the yellow, and the most growth or off the chart growth in the blue tubes. The exception here is going to be the fastidious organism, and I'm not sure which one it is. It's either the strep or it's aphacalis. Don't know which one you used. And in which case, you will still see more growth as you get toward the blue, but it's going to be far less than in the E. coli tube, right? So you're measuring the support of different nutrients, but you're also checking the fastidiousness of microorganisms. And that's always a good one. Let's see if I can spell this. Fastidi. I believe that's how fastidiousness is spelled. It's a couple sneaky eyes in there. Now, this is pretty easy. We do this a lot. You can do this for chlorophyll. You can do this at 600 wavelength, 600 lambda for just cell counts. So this is a really useful machine. And it does a couple other tricks and bells and whistles that we're not going to be worried about for right now. But the more money you spend, the more things that it does. And sometimes it's a really inexpensive but complicated machine that really is useful in the lab. Before these machines, and I even hate to bring back the olden days, thank the Lord it was before my time. Scientists used to actually have to sit down with one of these, and they would have to use a clicker, very much like what an umpire would use to keep track of balls and strikes, while they looked in a microscope at a slide that had grid patterns on it. And so these grid patterns are measured. They are very, very calibrated. And with each of these hemocytometers, you would get the calibrations so you knew what math to do. So the first thing is you would dilute it to something less concentrated, and then you would put the sample on the slide, look at it under the scope, and you would do cell counts in probably 5 to 10 of these individual grids. You would take the average, you would use a dilution coefficient to figure out how many cells per mil you have. I think we would agree the spectrophotometer, way better. But these are still used. These are still very low budget and labor intensive methods that we start a lot of undergrad and graduate students out on. It is also still very useful if you stay in your cultures or samples to do this in the hospital. It is just labor intensive. But the slide, maybe 50 bucks. The microscope, you already have. The samples, they're cheap. And so you can do a lot with a little. My personal favorite is stop doing this by hand. Let's let a computer handle it. And I have had the fortune of using something called a flow cytometer. Now, a flow cytometer needs to be calibrated, don't get me wrong, but you load a sample into a tube, and it's a very narrow tube. The idea is one cell per cell. That's a terrible way to say it. They line up in a single file line. That's a better way to say it. And as they go past an electronic detector, it measures using lasers how many cells pass the eye in how much time. And it's not just a number of cells, although that is very valuable. On the right, it also shows you how big they are. It gives you something about their size based on optical properties. So I can set it to just show size. And what we usually use is front scatter and side scatter. So if I have an E. coli cell, I've got my doodah here, right? This would be an example of side scatter. That would be an example of front scatter. So it would bounce the light off of it from different directions, and it would take measurements on how big it is. Some of these cells may fluoresce. So usually they come with a blue laser and a red laser if you have a fancy one. And by fancy, I mean you spent $300,000 or more. So they're not cheap. And you can plot based on size, fluorescence versus size, side scatter versus fluorescence. You have a variety of options to come up with plots like this. And so what you're looking at here is just size on size. 10 to the 0, 10 to the 1, 10 to the 2, 10 to the 3. These are probably in micrometers, so 10 to the 3 gets us to a millimeter. Same thing on this side, 0, 1, 2, 3. So these would be the largest cells out here. These would be the smallest cells down here. Now, the beauty about a flow cytometer is I can then draw a gate around these, right? I can map these out, or I can draw, with the program, a gate around this group of cells. And when I refeed the sample in, it'll pull these cells out and spit them into a separate vial. It'll put the rest of the cells outside of my polygon into the original vial. And so you can use this to separate populations also. Awesome device. Again, very expensive, but I would rather find a lab that had that over a lab that has this. All right, awesome. Yeah, I think, well, yeah, I guess it would be, right? So that you look for red cells, you look for white cells, you look for artifacts, right? So sometimes crystals, sometimes calcium particulates, those types of things. I could imagine it would be. The question is how many folks have access to the flow cytometer versus how many folks are doing direct comparisons using actual urine in a slide or blood in a slide, because let's face it, it was probably primarily designed for blood originally. Right? Before a machine, how do you think we knew how many erythrocytes and leukocytes and platelets everybody had? We had to count them. And I don't mean to laugh at that. It was a necessary job, but yeah, I think in CBCs and in certain other specialties, this has been replaced. This is far too fast and far too accurate. All right, let's see. What are some other things we should talk about? Yeah, I suppose we should talk a little bit about this. All right, so I'm going to click here quick, and I'm going to also hit this quick. So that would be the end of our chapter 7, or at least the nutritional piece of our march forward in examining prokaryotes. And you can see how there's a lot of chemistry in here and how a lot of chemistry ties into a lot of metabolism. And so the metabolism chapter is really big. And there's no way we could, or there's really no way we should cover it all. And I'm going to look at some faces while we're thinking, although the still pictures don't help. I'm assuming you talked about citric acid cycle and Krebs cycle and glycolysis, those types of things in A&P. Because you probably should have. If you took Gen Bio, I know you looked at them because we do photosynthesis and respiration there. I know I do it. If not, obviously, I don't want to hold folks responsible, and it's not the major point of anything. But when we get to biochemicals, some of those processes would be really helpful to review, especially for the lab, because you're going to be asked about glycolysis, right? So an organism that ferments glucose, right, you'll be asked to identify acid end products or not. Why would the products of fermentation produce acid end products? Well, because that's what they do, right? We usually reduce pyruvate to an acid, or we usually produce CO2, which is in itself acidic. And so the biochemical transformations from the Krebs cycle and those types of things, glycolysis, really are helpful in the lab. We can't get away from the chemistry, but more importantly, they illustrate the point that enzymes control metabolism. And in all of this, that's the big picture I want you to think about. Enzymes control metabolism. And so if an organism doesn't have an enzyme, then it can't do particular pathways. And so if an organism can't do a pathway, if you were to do a chemical fingerprint, you would be able to identify organisms based on the genetics vis-a-vis the chemistry, vis-a-vis the metabolism that they have. And so that's the link between the organisms and the biochemicals. The metabolism and the enzymes are super, super important. Right? You covered the nutrition piece, so you know what's in there. Right? Then we take a look at metabolism, and I don't want to spend a lot of time reviewing what hopefully is a review of enzymes, but I do want to talk about the highlights that are going to help with genes, the highlights that help with nutrition, the highlights that help with the lab. And that's really why I want to do this, so that I don't make folks nuts, because there's a lot of crazy words in here. But when we understand the sum total of all chemical reactions, it gives us a better picture of what's happening in the cell. Who done it? Who is our cell? What is an E. coli? Why do we care? What does it do to us? What makes it so good at what it does? And since there's no hearts, and there's no bones, and there's no ligaments or tendons or eyeballs or ears, all we can do is rely on what they have: ribosomes, genes, and gene products, right? We already looked at cell walls. We've already looked at plasma membranes. So that's really all that's left. How do these things regulate themselves? How do they reproduce? And what do they do to make sure that that happens? So the idea of biosynthesis, if we think about humans, is dynamic. It changes from time to time based on genes that are on and off. Sometimes we're building, so we're taking smaller products, inputting energy and making more complicated things like making proteins. Or sometimes we're hydrolyzing and breaking things down for energy, right? Do bacteria do exactly the same thing? Yes, they do. Exactly the same thing, right? But they're not using a liver to do it. They're not using a kidney to do it. They're not using nephrons or hearts or any of the parts that you would think of as a higher vertebrate, right? It boils down to, can the nutrient get into the cell? Yes or no? And if yes, is there an enzyme that can deal with it? Right? That's as simple as we can make this. It's Occam's razor. It's actually genius. And so when thinking about these enzymes, because enzymes are 150% the driver of metabolism here. I'm sure you've seen these before. It's definitely a Chem 105 topic, right? What are enzymes? So I don't know if folks want to go around or they just want to kind of look through this chart real quick, right? I always start with thinking about them as globular proteins, right? And as we know, proteins have to fold to be active. So totally true here. Some of them have cofactors. They might be metals, that might be other proteins themselves, smaller peptides, that help them configure and or help them catalyze reactions. One of the common ones that we talk about with Krebs is TCA, right? Krebs TCA cycle is our coenzyme A. So we'll have acetyl-CoA. And so that coenzyme helps bring in the carbon to the Krebs or the citric acid cycle. They are organic. So all they do is speed up the reaction by lowering the activation energy. So they're not changed, they're not used up, but they do lower the barrier to activation. What do I mean by that? Well, we're talking about the rate. And so I studied urease. I can talk about urease metabolism. A molecule of urea without the enzyme urease does not degrade for 3.5 years, all things considered equal. It's very stable, and it takes a long time for it to spontaneously degrade, 3.5 years. If that one molecule of urea is acted upon by an enzyme, a unit of urease, we take the time to hydrolysis from 3.5 years to 1.5 microseconds. 3.5 years, 1.5 microseconds. So that is a great increase in speed at the cost of the energy to make the enzyme, right? So that's the trade-off. Most of these have what we call an active site. And generally it's one peptide per substrate. So it's one enzyme per substrate in general. So urease works on urea, citrase works on citrate, so forth and so on. Lipases, and that's a funny thing, lipases work on lipids. Which lipid? Well, it depends on which enzyme is going to come to the party. And normally, as you probably heard, they end in ASE. So if you're not sure it's an enzyme, normally you'll see it written above the equation arrow, but normally it will end in ASE, aside from things like pepsin, which are enzymes that don't end in ASE. Generally, they're massive, right? So an enzyme to substrate ratio, 50 to 1 would not be uncommon, that the enzyme would be that much larger. They do bind to the substrate, but they're not used up, they're not changed. And we can recycle them even in low concentrations. Different enzymes have different affinities for substrates. Different enzymes may pull or act on substrates with different rates. That's totally true. And enzymes or enzyme systems can become saturated. All right, so what that means is we're used to adding more substrate to see a faster reaction. And so at the beginning of an enzymatically catalyzed reaction, the more substrate means you'll see faster reactions because there's free enzyme. At some point, we hit an asymptote where more substrate does not generate a faster reaction. If you want a faster reaction, the body has to make more enzyme, which is energetically expensive. A microbe has to make more enzymes, very expensive for such a small critter. And so generally, there's a rate of reaction where it tops out at, and it does not or is not exceeded by. So they're not permanently used up, they're not changed, but there's also only so much of them in a cell. So we recycle them, and their activity is heavily biased on availability of substrate, temperature, pH, and usually we throw salt conditions into this, right? Because any of those conditions, any of those parameters can cause an enzyme to denature, unfold, if they're not in the optimal physiological parameters, right? And obviously feedback, negative feedback usually is what turns these cycles on and off. And we can do this at a protein level by inhibiting other enzymes, or we can do this at the gene level by turning off the gene. And the tryptophan operon is usually a really good one to look at if anybody's wondering. So there's a lot of stuff in here. I just want to take a look at some of this, and we're probably not going to get too crazy here. There are a couple different kinds of enzymes. There's a bunch of enzyme classes. So I'll show those to you briefly. We'll talk about constitutive versus non-constitutive. We'll look at a couple classes. And I think that will probably be more dense than our heads want to deal with tonight. So enzymes might be simple enzymes. And by simple enzymes, I mean one strand, one peptide. Sometimes enzymes are multiple subunits. And so like my urease enzyme was 7 subunits. It needed to transcribe and translate urea A, urea B, urea C. That's the structural piece. So it was a trimer. And then I also needed urea D, F, E, F, G, and H. D, E, F, G, and H, I'm sorry, to make all of the accessories that urease needed to hydrolyze urea. So A, B, and C were just the structural enzymes. D, E, F, G, and sometimes H or R were the accessory and regulatory components that go along with the enzyme, which I'm going to admit is ridiculous. But we know proteins aren't always just one strand. And I'm not suggesting that hemoglobin is an enzyme, but certainly it's a protein with multiple subunits. So that is common, and we did see that even in A&P. Some of these enzymes, though, aren't just protein. All right, some of them have an apoenzyme, so that's the protein portion, and a cofactor. A lot of the times it's a metal. Sometimes it's a vitamin or an organic molecule, but most of the times it's a metal. And again, thinking about this, the heme group in hemoglobin is an iron. The porphyrin ring of chlorophyll is a copper. Magnesium, tungsten, chromium, a lot of these trace metals end up as cofactors in a lot of our enzymes, and that turns them into what we call metalloenzymes. And so urease, in fact, does require nickel. And so urease is what we call a metalloenzyme. And so without nickel, it doesn't work. Sometimes too much of these cofactors actually disables the enzyme, right? So iron, copper, magnesium, we know they participate in redox reactions. And so sometimes the exchange of electrons or iron in the +2 versus a +3 state might be toxic. Copper +2 instead of +1 might be toxic. And so when we make media that includes these weird trace metals as a cofactor, sometimes we don't have everything we need on hand. Sometimes we have to substitute. And I was always told, and this has worked well for me, if you substitute a metal, make sure it's in the same redox state. So if you have, I don't know, let's make something up. Instead of iron chloride, if you had iron bromide, probably would work fine, as long as iron was the same +2 in both cases. If one of them was +3, one of them was +2, you might be making a mistake. So just for what that's worth. And then organic molecules, vitamins, sometimes act as cofactors. We see this in AMP. We see this with microbes. And it may look something like this, right? So basically this metallic cofactor, a coenzyme, or both, bind to these hybrid enzymes where it's the protein and a cofactor in order to get them to work, in order to have the active site or this cleft that you see, it's probably this one, the cleft that you see in these enzymes. So without the metal, it doesn't fold right, it doesn't work. Without the cofactor, the coenzyme, doesn't fold, it doesn't work. And so that's a common theme here. And obviously we don't want to bother people with Monod kinetics, Michaelis-Menten kinetics. We don't need to care about any of that stuff, right? But just kind of keep on the form and function relationship and the idea that these need to fold to be active. Just a couple looks at enzyme substrates here. The substrate needs to fit in the active site. So obviously the one peptide per substrate is important. This one fits, this one will be metabolized, right? And we see the products. Notice the enzyme is unchanged. This one will not fit, so it stays unchanged unless there is a separate enzyme to do the job. This one here, you're seeing more of an induced fit mechanism. So the enzyme fits into the active site, and then the active site changes around the substrate so that the reaction can be catalyzed. Kind of cool, right? So that lock and key and induced fit method, both of them come into play. I've mentioned a lot of things about enzymes, especially with staph. We talk about exoenzymes versus endoenzymes, right? So staphs make heat-stable exoenzymes. The key here is exo, outside of the cell, where they break down a lot of things, right? Hopefully for food, but sometimes they make exoproteins that act as toxins. If anybody in the room has had the staphylococcal food poisoning, you know all too well what an exoenzyme, or in this case, the exotoxin in staph, what it does. Vicious diarrhea, vicious vomiting, chills, fever, and the emptying of anything that's been in your body for days. Right? If it stayed in the cell, it would be less of a problem. It would be handling the metabolism in the cell, but instead it's released and you see the harm it does. Notice some of the ones here. Cellulase is an exoenzyme. Amylase is an exoenzyme, so it's actually diffusing out of the cell and breaking down sugars in the cytoplasm or in the culture medium. Notice penicillinase is a good one, right? So this is a beta-lactamase, if we're going to speak a little more properly. And so the cell doesn't actually bring it in to destroy it. The cell, if it's able to make this and has a plasmid, releases the beta-lactamase into the media to destroy the antibiotic before it gets near the cell. And in thinking about it, it's a really cool strategy. So in the lab, if you're shown a starch plate or a tributyrin plate, right, which are both macromolecules, one's the polysaccharide, one is a triglyceride, you're going to see exoenzymes released by the organism, and you're going to see a halo or a clearing around the organism in a media to show that the enzymes have been working, to show the enzymes have destroyed the oil in the tributyrin or hydrolyzed the starch in the media. And you'll test for starch by using iodine, right? So some of that stuff is still tried and true. What are some other things in here we should talk about? Right, these are definitions, but these are worth looking at. The idea of constitutive enzymes versus induced enzymes. And so let's see. Maybe the better way to think of this, or maybe not, is the idea of housekeeping enzymes, ones that are always around, that handle your basic metabolic functions, involved in the things that you need to keep yourself going every day. Or in a microbe, it's largely glucose metabolism because it needs to eat pretty much all the time. So these enzymes are always on, and while feedback may turn them off from time to time, right, these are generally on unless you turn them off or unless an organism shuts them down. Remember, enzymes are complicated proteins. It costs them energy to make them. And so if we don't need them all the time, we're not going to produce them all the time. That's where the induction comes in. So enzymes we induce are there in genetic code, just hanging out. And when there's an upregulation or when we turn the gene on, we'll talk more about that next week, then the organism transcribes and translates the enzyme. Sometimes the controller of this is a strange environmental compound, a weird sugar, a weird protein, or an abnormal metal. And that's what turns on the enzyme pathway that had been off. It says, hey, we need it. Hey, you could use this, or hey, you better do something about this before you're dead. Right? And in the case of penicillinase or the beta-lactamases, those are induced until you expose an organism to it, and then they're constitutive pretty much for the rest of its life. We don't tend to see antibiotic resistance go away. And that's an unfortunate piece of the puzzle here. In terms of, let's see, how many more things do I want to show you? In terms of the kinds of enzymes that you see, there are 5 or 6 big classes. And generally, we break out oxidases and reductases into 2 separate classes. And since scientists aren't imaginative at all, the names should say it all, right? So oxidases steal electrons. Reductases add electrons. Pretty straightforward. Aminotransferases move around amino groups. So we're taking the NH2s and we're swapping them around. It doesn't change the R groups. It just moves around the amino groups. So that's kind of important here. Phosphotransferases. I often don't like that word. A better word maybe that you're familiar with would be kinases. Right? Kinases are enzymes that move phosphates around or that phosphorylate. So we don't call them phosphorylases, unfortunately. We call them kinases. But the larger group here is phosphotransferases. We're moving around phosphate groups. So you'd see them in ATP. You would see them in substrate level phosphorylation. You'd see them in oxidative phosphorylation in the electron transport chain. Methyltransferases move around CH3s, so that's pretty self-explanatory. Lactase. What about lactase? I don't know where I caught or which one I missed here. So let's see, lactases are hydrolyzing lactose. So if we're hydrolyzing it for the purpose of energy, we are probably oxidizing it. I guess that's where we're going, right?
I just, from induced enzymes, like that we don't need all the time.
So being a digestive enzyme, I guess if you're someone who doesn't, I don't know. I don't know that it is I guess it probably is, but it's not one that ends up in a deep sleep. I don't know that we're not producing it at basal levels. So I don't want to say one way or the other. Urease is one of those that's always produced too, so that's not a good example. Bacterially, I can tell you the lac operon is induced, whereas like the tryptophan operon is repressed. but I'm not sure. Let me take a look here and see, because now you got me curious. And that's one of the benefits here. We can actually look stuff up. Lactose induced. Lactase activity starts during the third peak. How lactase activity post weaning? Let's see. Lactase persistence. This is a ridiculous article. So I just read a quick article. I'm going to throw this in there. Just maybe this helps to answer the question or not. It seems like lactase activity peaks when we're children, and that there's a few control mechanisms and/or with the help of lactose metabolizing organisms in our gut, maybe it keeps some of us out of trouble. So it seems to say that it's produced and the level is set as we're children or when we're children, but I'm not going to try and read that whole thing right now, but that might be helpful to you to see that. Generally, what I think about lactose intolerance isn't at the organismal level. I think of it at the heredity level. So if it's not something you use, you typically lose. And I'm thinking about the Far East and populations where animal milk is not typically consumed. So over thousands of years, folks on the Indian subcontinent, China, Japan, Korea, Vietnam, that part of the world, generally lack the gene for lactase, which is why it's not there. It's not so much a transcription translation issue, it's a presence or absence of the gene. All right, so maybe the paper describes a little bit more how it's upregulated and how it sets up, but lactase is probably a little more complicated because it does mimic patterns of heredity. But there is that use it or lose it piece, so I guess I have to say that. And in terms of enzymes, it's probably an oxidase. But the last one is a decarboxylase. And so, the article's good. Okay, good. Decarboxylases are moving around a CO or CO2. And there's an enzyme called Rubisco that maybe you've heard of. I'm hoping you've heard of. Maybe not. Right, it's part of the photosynthetic light-independent reactions. And so Rubisco is ribulose 1,5-bisphosphate carboxylase oxidase. So half of the time, Rubisco removes a carbon dioxide or transfers a carbon dioxide. The other half of the time, it oxidizes. It steals electrons, which Rubisco is a terrible enzyme. But a lot of the times you see these carboxylases, these decarboxylases, the movement of carbon dioxides is very, very common, especially in the glycolysis pathway, especially when we respire away carbon. So you'll see a lot of these in energetic pathways, honestly, just in things that metabolize biomolecules of carbon. So like most things. How much more do we want to say? All right, a couple more things. Enzyme activity is important to control. And generally the feedback inhibition, feedback repression get at operons. So something is on, a gene is on, it gets to a level where the gene product is too high, and so we simply switch off the pathway. And so in one way, we do this at the protein level. That's the inhibition piece. At the repression piece, we do this at the genetic level. Now, enzyme induction could be, enzymes are only made when substrates are present. And I'm not sure if that article talked about lactose inducing lactase or not. It seemed, I didn't read it very carefully, but it seemed to indicate other. Urease could be induced, but typically it's at a basal level, but there are some enzymes like this. The more interesting piece of this is competitive inhibition. Substance that resembles the normal substrate competes with feedback for active site. All right, so this is an interesting one. And I guess before I get going, is Mario still around or is Mario gone? Did he have to take a call? And I'm only asking him because he's at Spring Hill and he's a medic, I think. It's not an EMT, but a medic. I guess he's not around. All right. Oh, he's here. So talk to me about what happens when, and I'm going to mention antifreeze and consumption here. Talk to me about what happens when you find a patient that's drank antifreeze. Right. I know there's some new school methods, but do you remember the old school method that we used to talk about back in the day, maybe back in Bio 106? Right, and it comes under the realm of competitive inhibition. So we know that antifreeze is polyethylene glycol, right? And because it's a glycol, it's got an alcohol group on it. It ends in OL. Since alcohols are metabolized in the liver with a set of enzymes, oxidases, dehydrogenases, aldehyde dehydrogenase being one of them, the metabolism of antifreeze happens in the liver. And generally, that's got a very negative outcome for people who drink antifreeze and are not found. For animals, it's very dangerous because there is no antidote for a cat or a dog that consumed antifreeze. And so essentially, they tend to die. In human beings, and maybe Mario's still typing, I don't want to cut him off here. There are now medications that block the enzymes that would metabolize the alcohol in the liver. And these are a relatively new invention, probably in the last 20 years, because it happens enough, it was worth producing. But in the old school days, an EMT or a paramedic who found somebody who was suspected of drinking antifreeze would typically stop by the liquor store on the way to the hospital and pick up a liter or a handle of vodka. Now you're thinking to yourselves, did he have that much of a bad day that he needs to drink? No, not for the medic. It's actually for the unresponsive patient. So the enzymes in the liver are working on the substrate that fits into the active sites. Polyethylene glycol, or antifreeze, is a pretty big molecule. Ethanol is a much smaller molecule. And so if you load up a patient with more alcohol, ethanol, than they have antifreeze, the enzymes that break down alcohol will go after the ethanol and it will leave the antifreeze alone. So you're blocking the enzyme from a pathway that it shouldn't be doing by giving it a substrate that it would rather work on. And so that is just straight thermodynamics. Ethanol is a lower molecular weight. Your liver will process that preferentially than the antifreeze, giving the medics in the hospital time to get rid of the antifreeze, dialyze it away, active charcoal it, get you to throw it up, pass it, whatever they have to do to get it out so that it doesn't form those solids and destroy the liver. Right? It's an extreme example, but it's a pretty old school example of why the medics used to do that. Right? A little bit of experience and a little bit of knowledge really goes a long way here. All right. What else do we want to say here? Yeah, there's probably plenty on the way to Nyack Hospital. I guess it is good to know. Although they might look at you like, I don't think you're old enough to know this stuff. I wonder where that came from. That's good. That's too funny. It's serious, but it's still kind of, yeah, okay. Should we say anything else? Most of what else is here, respective of enzymes, and it's only the stuff I'm showing you that I'm going to ask you to remember for the test. Most of what else is here probably is stuff that we remember. But you know what? There's two things that are, there's one thing that's worth looking, right? And that's just the idea of redox reactions, because I asked this in the Chem 105s and it's a little dicey. I asked this again, or it's asked again in A&P, it's a little dicey, right? And we ask it again because you need to figure it out for micro. So the idea here, redox reactions are coupled oxidation and reduction reactions. So wherever something is lost, you must gain. So if I oxidize something by stealing electrons, I've got to reduce something by giving the electrons to something else. I can't just sweep them under the rug. Now, it's really, if you sit there and think about it, what the electron donor is, or we call it a reductant sometimes, or what the electron acceptor is. And so when you look at it, sometimes we're just trying to see, where did the charge get reduced? If you can see where the charge was reduced, you know which molecule was gaining electrons. It's real easy with salts, it's easy with acids, it's easy with bases. Most of the other compounds that we look at tend to be organic in nature. And so using the idea of pluses and minuses is kind of useless because of their nonpolar nature. And instead, what I like to suggest is you see an oxidation, you're looking for the creation of a double bond. When you see a reduction, you're looking for the creation of single bonds or the breaking or converting of double bonds to single bonds. Why is that? Well, when a double bond appears, they have to share more electrons because there are less of them. When a single bond appears where a double used to be, it means that there are more electrons and they don't have to share as many. So you can use that as a guide for oxidation and reduction and for things that are not charged. And obviously, and I know this is the problem, I have to think about it. Leo goes, GER is a good way to remember it. Loss, electron, oxidation, gain, electron, reduction, right? Really simple. The other one is oil rig. Oxidation is loss, reduction is gain. And so if you're not sure where to start, say it out loud, right? And you're looking for where electrons are lost and where they're gained. All right, here's a true or false, and I might put this on the test because it's a perennial problem. Oxidation has to do with oxygen. All right, so 50-50 here, true or false? Either we stepped away, oh, okay, there we go. Here we go, good, 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 good. Right, an oxidation is a loss of an electron. Doesn't matter where it goes. Right, now, in the body, would it be oxygen? Yeah, most likely. But chemically, an oxidation is a loss of one or more electrons to any electron acceptor. And so a lot of the time it is oxygen, yes, but it doesn't always have to be. Sometimes it's nitrate, sometimes it's sulfate, sometimes it's carbon dioxide, sometimes it's methane. Really depends on the organism and the system that it's in. Sometimes these are lost to electron carriers. And if you read a text about plant physiology, and I know it's boring, but it's a good place to go. If you read a text about microbial metabolism, if you read, it's false just because it doesn't have to have anything to do with oxygen. It may, but technically oxidation is a loss of an electron. It may have nothing ever to do with oxygen. Sorry, that's just in the chat. The electron carriers that I'm referring to here, we would tend to refer to as reducing power. So from your knowledge of glycolysis, right, when we break down glucose into pyruvate, and I probably have it, I should just show this to you. Let me see, here we go. And I know this is gonna bring back bad memories. Right, when we take glucose and we break it into G3P here, right, notice we use ATP, right, so we're using energy. But when we take the pyruvate and the G3P and turn it into pyruvate, notice these weird things pop up. NAD, NADH, NAD, NADH. That's an electron carrier. NAD plus is oxidized, NADH is reduced. Now, what happens to all of that NADH that we make in glycolysis and in the Krebs cycle? Well, you know it joins its partner FADH2 in the electron transport chain. So in this piece, in the energy payoff piece from G3P to pyruvate, what we end up doing is transferring the electrons from the G3P, from the carbon-containing compounds, so they're oxidized, and we reduce the NAD+. So law of conservation is good. And in general, what we're generating is something called reducing power, right? The ability to add electrons to a substrate later on. Those substrates happen to be the cytochromes, but it doesn't matter. And so sometimes it's not just, oh, we oxidize an iron and we reduced an oxygen. Here, we're oxidizing a carbon-containing molecule, and we're reducing an energy carrier. We're reducing something that can reduce other stuff later. And so the reducing power here ends up going from glycolysis and the citric acid cycle all the way over to the electron transport chain. Notice NAD is here. Notice FADH is here. We oxidize the NADH back to NAD+, and we start reducing now FMN, coenzyme Q, cytochrome B, C, C1, A1, A2, A3, in the process of converting that reduced energy in the form of electrons to chemical energy in ATP. So yeah. I'm not going to ask you to recall stuff we didn't look at. I'm not going to ask you to read the entire chapter 8 because it's a lot. I think a lot of the lab will help reinforce that. It should bring back memories of AMP and glycolysis. Hopefully it does, especially when you look at the mitochondria and the mitochondrial matrix and chemiosmosis and the proton motive force and all those terrible words that you're having flashbacks with right now, including hypertonic and hypotonic and isotonic from earlier in the evening. So yeah, with that, I'm going to hit stop.